It's a great pleasure to see you all here this evening. I'm going to take you on a rather strange journey, and it's a journey which is my own, really, and that of my research group over the last 20 or 30 years. But before that, I'm going to just set what we do against a backdrop of the last 300 years or thereabouts in thinking in biology. So if you go back nearly 300 years, I think there's been something like 5 or 6 major revolutions in biological thought since then. The first, arguably, was the invention by Linnaeus of a way of naming organisms, what's known as binomial nomenclature. And that's the system we still use to name and classify the diversity of organisms on the planet. Explaining that diversity fell to Charles Darwin. And in 1859, he published his famous book on the origin of species and gave us an explanation for the abundance and diversity of life on this planet. Then, the next revolution was what's called neo-Darwinism, which saw a conflation of Darwinian theory with the science of genetics. And that, towards the middle of the century, led to the birth of the molecular era. And you could say that that began in 1953 with Watson and Crick's description of the structure of DNA. Now we're in the fifth revolution at the moment, and we might call it, and many do, the epigenetic revolution, where the focus is on how adaptive phenotypes, and by phenotypes I mean the way that organisms work and are, how they arise during their development as a result of plastic interactions between genes and the environment. And that's a huge area of research which is really exploding at the moment, and it's helping us to understand how genes play out through development to yield complex phenotypes. But the next revolution, the final frontier, if you like, in biology, is to take that understanding all the way through to the way that ecosystems work. So if you consider phenotypes of individuals, they are members of groups and populations which in turn comprise communities of different species and ultimately ecosystems. And it's not just a simple linear hierarchy, the whole thing's girded by abundant feedbacks. And if we're to understand that complex relationship, a web of interactions, where do we start? And I think I'm going to argue tonight that we need to start, and I hope I'll demonstrate this, at an understanding of the phenotype. And the reason is that it's natural selection that sees the phenotype. The phenotype is visible to natural selection. So selection sees the speed and maneuverability of a cheetah, not the muscles and genes and neurons that go to make up that behavior. It's the outcome of the behavior that's visible, the way the thing works. All right, with that backdrop, I'm now going to take us to the midst of a swarm of the desert locust in North Africa. Now, this animal is one of a dozen or so species of grasshopper that are called locusts, and we have at least one species that's of significant agricultural importance in this country, and I'm sure you've heard all about the coming locust outbreaks. Now, these species of otherwise normal grasshoppers are united by one defining feature, and that is that they're essentially two animals packed within the same genome. So let's take these two juvenile desert locusts. They are sisters. They were born from the same pod of eggs laid by their mother. The insect on the left, she was reared on her own from the time that she hatched from the egg. And you'll see that she's grown up to assume this rather beautiful green colour. She's well camouflaged in her habitat. She would be very shy in her behaviour, and she would avoid other locusts, except when she was a mature adult and ready to mate. In contrast, her sister here is brightly coloured. She was reared in a crowd. She is also highly active in her behaviour. She's attracted by other locusts and in large numbers would contribute to massive marching bands of juveniles, and when they become winged adults, huge swarms of winged adults that can fly hundreds of kilometres a night and can comprise billions of animals which collectively over a single day would eat hundreds of thousands of tons of vegetation, and that's why they're such a problem to humans. Now they're strikingly different, and this change is occasioned by population density, whether or not you're crowded. But the very first thing to change in locust species upon being crowded, if you take solitary animals, is their behaviour, not their appearance. That takes some while to change. Their behaviour, however, can change within a matter of an hour or less. So if you crowded this animal here, within a short period, she would be, rather than repulsed by other locusts, she would be attracted by them, would be aggregating with them and contributing potentially to marching bands of other juveniles. So that's an extraordinarily fast response to a change in population density. And one of the very first questions we wanted to answer was, well, what is it about being in a crowd that causes that shift? Well, if you think about it logically, it could be the sight of other locusts, it could be the smell of other locusts, perhaps being physically in contact with others, or perhaps there's sound. And I won't go into the details of the experiment, but in an early experiment we teased those things apart, both singly and interactively, and found that a key stimulus was touch. Now, a locust is richly endowed with touch-sensitive hairs. They cover them all over their body. And we wanted to know whether a particular part of the animal was the key part which was responding to being in a crowd. And to answer that question, we resorted to an extremely high-tech piece of scientific research. It's called a paintbrush. So we sat, and I spent many, many hours in a hot room stroking locusts on different body parts for 5 seconds a minute for 4 hours, at the end of which we assayed the behaviour of the animal and found that extraordinarily there was only one body region that was effective, the hind leg. Now this is the North African species that I've been talking about. Very recently in the lab we've done the same experiments with the Australian plague locust, and I think really excitingly in that case it's the antenna that provides the source of stimulation. So different species have ended up with the same outcome, locust-like behaviour, through different means. But anyway, I'm going to focus on this animal here now, the North African species. Now, having localised the stimulus, the site of stimulus to the back leg, that was very fortunate because it opened a window on the underlying neurochemistry of the process. We could target this back leg and ask the question, well, where do the nerve cells that innervate the hairs on that back leg and the armpit, if you like, of the back leg, where do they send their projections into the nervous system? And can we start to trace back the neural and neurochemical mechanisms of this extraordinary behavioural change? Well, the first thing to do under those circumstances is to start an experiment where you lay the animal on its back, you expose the nearest bit of the central nervous system. Now, insects have a chain of brains along their body, the ventral surface of their body. This is the nearest one to the hind leg. And then by electrically stimulating individual nerve branches or cutting them, we're able to focus in on the particular pathways carrying the information, which was telling the animal, in a sense, that it was in a crowd or not. We found that there were indeed particular beds of hairs here and particular receptors in the armpit of the animal that were carrying the critical information about population density. Now that information's going into the nervous system and somehow evoking a really radical change in behaviour. The animal's no longer repelled by others, it's attracted by them. If you think about it logically, because that's happening so quickly, it can't be because there's been a physical rewiring of the nerves in this part of the circuitry of its nervous system. It has to be something more transitory, something probably induced by chemical modulation. So that allowed us to start looking within this region and to try and assay the number of different potential or putative modulatory chemicals and see which ones showed the appropriate time course, and then to use pharmacological manipulations to try and manipulate their levels and see whether we could identify which chemicals were critical for the change. And to cut a very long story short, the answer was that a single neurochemical was important. That was serotonin. Now, serotonin mediates this change. It's both necessary and sufficient. You can mess with its levels in that part of the nervous system and you can cause the change in behaviour to occur, or you can block it. Now, serotonin's not just found in the nervous systems of locusts, it's found in our own nervous system, and indeed the nervous systems of all animals. And, as you probably know, the serotonergic pathways in the brain are involved with arousal, social behaviour, and a whole series of obvious behavioural phenomena in us as well as in other animals. And, for example, some of the antidepressant medications act on that pathway, and so too does the drug ecstasy, which led, of course, to the typical newspaper responses and media responses. Plagues of locusts in ecstasy, bugs on drugs was another of my favourites. And I also liked, because this provides us with a little forward look, there's a little chubby person here, and that's a link to something I'm going to talk about a little bit later. So we've identified the nature of the inputs. This is how the animal's detecting whether it's in a crowd, and the mediating chemical agents. The next step, and this is work now being conducted by my colleagues in Cambridge, is to start tracking which individual nerve cells within that part of the nervous system are the critical ones. Because we know we can map which nerve cells release serotonin. We can ablate and record from each one of those and start to focus in even further. Perhaps there's a single pair of nerve cells, responsible for mass outbreaks of locusts in North Africa, or indeed in Australia. Well, I'm going to stop at that point. It's given you, I hope, a taste of how you can take a behavioural phenomenon and work it through down, in that case, to a biochemical, neurochemical answer. And indeed, and this is one of the other major aims of the lab, to take it even further to the expression of genes that embody then that behavioural change and start to express it in other ways within the animal's body. So we can start looking at the molecular and genetic mechanisms as well. But I'd like to turn now to an ecological question. Why do locusts, solitary locusts, come together in the first place? Because they don't like being together. They avoid one another. Why on earth do they come together in the first place? Well, the answer came from using a series of simulations where we manipulated the number of locusts, the amount of food resource and the distribution of that food resource in the computer, and tested those models both in the laboratory and in the field. And what we found was that the critical thing is the distribution of resources in the habitat. As resources become limiting and clumpily distributed, that clumpiness encourages the locusts to have to come together despite their predisposition to avoid one another. And in that way, as soon as they're together, they start jostling, and that triggers the change. They become attracted to one another, and you can start to see or get a feel for the explosive nature of the process thereafter. And as good evolutionary biologists, we can also ask the question, well, what's the point of showing this change? What's the function? What's the adaptive significance? And there are several reasons, and I won't go into them in great detail, but there is an argument for safety in numbers. When you're in a beyond a critical number, you might as well clump together rather than disperse, because you're diluted by others around you. And there's also, through use of a body of theory which is called percolation theory, an argument that by clumping together in superaggregations, you hide yourself in an otherwise large landscape. The same way, for example, as the North Atlantic convoys stuck together during the Second World War to avoid U-boat attack. So what we have then is an environmentally induced congregation of animals that would rather avoid one another. They then flip into an active aggregative state, and they're all together milling around. And then something extraordinary happens. They're going nowhere, and then suddenly, as if of a single mind, the entire group of potentially millions of animals starts to move. It starts to move collectively, with all the individuals in that aggregation marching together in parallel. Why on earth do they do that? Well, If you look at them, they can cover hundreds of meters across, kilometers long, and they flow through the environment like a river. That sudden transition from disorder to ordered movement could be because there are some sort of hierarchy of leaders amongst the number of locusts. Some of them, the leader locusts, start to move and others follow, and away you go. Or alternatively, it could be just something that happens as a result of the interactions between individuals that are essentially all the same. Well, at that point, we turned to our colleagues in statistical physics, because they've been playing for many years with a set of models called self-propelled particles models. And what they are is you simulate in a computer individual particles that behave according to simple local rules. You do things in accordance with the behaviour of your neighbours. And then you see and if anything interesting happens at the group level when you consider the whole cluster of particles. And from those studies, we found that the collective behaviour at the group level, that sudden transition to collective aligned movement, emerges from a simple local interaction rule. All the locusts are in effect the same, and they follow a single simple rule, which is align with your moving neighbours. So just to show you an equation, this is a simple self-propelled particles model. It comes from the work of Thomas Wischek, published in 1999, and it's actually very simple. A particle, or a locust, has a position which changes as a function of its velocity, its inertia, its tendency to continue in that same direction, its local alignment, the extent to which it aligns with neighbours within a certain distance, and there's some sort of noisy term that you always find in biological systems. Now, what we did was we started to apply, and again, I'm using the royal we here, colleague Jerome Boole in the laboratory has done the majority of this work. What we found was that when you took this model and parameterized it appropriately, it could produce strongly locust-like behavior. But to do that, you need to commence a dialogue between the mathematics and the animals themselves. You need to be able to parameterize and test your predictions using real locusts. Now that poses a problem because when you have a locust marching swarm in the lab, it tends to alarm one's colleagues. So what we had to do was find a way of confining them so that they would go marching around happily but not go anywhere. And this isn't a fried egg with flies all over it. It's actually our patented Mexican hat marching arena. So what we have here are locusts marching endlessly round and round this arena, and using software that colleague Ian Cousin, now at Princeton University, who's in the lab in Oxford and spent some time here as well, devised. So we can follow individual particles, we can quantify their interactions, we can quantify collective properties of the group, the degree of alignment and so on. And using those sorts of models and then going out into the field, work we did here in Australia, to try and use real measurements of real marching locusts, and Jerome spent many happy months recording real interaction responses between 70,000 individual locusts in marching bands recorded in the field near Broken Hill. Having done that, we're able to then construct realistic mathematical models, parameterized models, which can then be used to predict how the entire collective will behave. And it's important to know when a group will start to march, in what direction they'll go, whether they'll shift direction, and how quickly they'll go, because that allows our colleagues at the Australian Plague Locust Commission, with whom we work, to devise better ways of applying chemical and biological control agents. So what I'd like to do now is just show you some of Jerome's very recent simulation models. This is a model in which he simulated 250,000 locusts over a 4-hour period. Those locusts have a soft repulsion within a 3.5 centimetre range. They align with others within a 13.5 centimetre range, which is exactly, we know, the distance that real locusts do align with their neighbours within. It has no attraction, and it has probabilities of the animals hopping and stopping to feed and so forth. So it's realistic, but nevertheless simple. And what I'm going to show you now is what happens when you've got such a simulation, 250,000 simulated locusts, starting in one place and then moving together following those rules over time. This is obviously greatly sped up. Now what you'll see there is it's rather like a comet. It's shedding locusts off the back, or simulated locust particles. It has a crescent-like front with a very steep decline in density from the front further back. And that happens to fit very neatly the sorts of behaviour we see in the Australian plague locusts. So what we're now trying to work towards is a generic model for predicting locust band movement. That's the mathematics. It was founded on real biology, but we need now to go into the field and test it. And to do that, we've started a collaboration with the Australian Centre for Field Robotics here in the University of Sydney, and they have devised a beautiful system. Actually, it's not going to involve helicopters. It's going to involve fixed-wing unmanned aerial vehicles. But they've devised a system which has a stroboscope on board which flashes, and then reflecting back from crystals that we mount on the backs of a certain number of test animals that we release into the swarm, we're able to use these fixed-wing aircraft to track and individually monitor the movements, and hence the interactions and diffusion rates and so forth between our test animals. And because the models make really quite clear diagnostic predictions about what should happen if you release animals into certain parts of a swarm, then we can test that using that equipment. We're in about 6 weeks' time to go into the field to test fly and to start running these experiments. So this is very current work. But it begs another question. Why on earth do locusts align with their moving neighbours? What's the point of that? Well, the answer to that turned out to be rather sinister and came from studies that we did. Colleague Greg Sword, who's in the department here, and I, along with Ian Cousin and Pat Lorch, had a tremendous amount of fun in Utah and Idaho over 2 seasons working on this thing. It's called the Mormon cricket. It's not a locust, it's a large flightless cricket. And it forms these massive aggregations. So in fact, this one here crossed that road continuously for 5 days. So you can imagine how big they are. They go for 1 to 2 kilometres a day. Why are they called Mormon crickets? Well, that goes back to the Mormon pioneers. When they first arrived in the spring of 1848 to what is now Salt Lake City, they looked down from the Rockies, having suffered terrible privations as they crossed the continent, saw this lovely lake and verdant green surrounds and thought, well, we're in paradise now. Turned out to be a hypersaline lake with salt vegetation, so it was pretty tough. Planted their first crops. They were just coming to fruition when over the hill came Mormon crickets and devoured them. So they beat them with sticks and flames and hopelessly tried to stop their food from being eaten. And then suddenly out of the sky descended flocks of seagulls, ate the Mormon crickets, saved the Mormons, and now the seagull is the state bird of Utah. Well, we wanted to know why they're marching. Is there something about their environment which has let them down, forcing them to continue to move through it? And they don't strip the environment bare as locusts do. They're actually rather more selective. So what we wanted to know was whether they're specifically deficient in any nutrient. So that involved setting up a simple experiment where we put dishes of synthetic food in front of the marching band. And these foods, there were 4 different sorts. There was one that contained both protein and carbohydrate, another that contained only carbohydrate, one that contained only protein, and one that contained neither protein nor carbohydrate, just the cellulose and vitamins and salts and other stuff that are in all of them. Watch what happens. So Mormon crickets have never seen these things before, obviously. They're marching through the environment. They come across these dishes, and bear in mind that they taste with their antennae and their feet. And that's a fairly nasty thought in my case at least. But they make assessments chemical assessments of their food by doing that. And if you watch over the next 10 or 20 seconds, you'll start to see something interesting emerging. It's protein. They're stopping at the protein. And if you do this, repeat it multiple times, and you do it in different bands and all the right things as a scientist should do, you find that their probability of stopping at a dish is directly a function of the protein in that dish. Don't care about carbs, it's protein. We showed that the crickets are actually on a forced march to find protein. And the reason they keep moving is because if they stop moving, they get eaten. It's a forced march for protein driven by cannibalism. Because the most abundant source of protein in the environment is another cricket. And many times people have said to me, oh, that's a very good analogy for life and academia. It's the same thing.